Hello. Today we're going to talk about muscles and muscle tissue. This is a long PowerPoint, so I'm going to split this into two parts for us. So your muscles actually make up about half of your body's weight. That's quite a bit. But your muscles can also do some pretty cool things. They can take chemical energy, ATP, and produce movement. They take that ATP and allow you to walk, run, write, read, sit, all of that. So we're going to look at the different kinds of tissue, muscle tissue, characteristics, and the function of muscles today. Whenever you see myo, mys, or sarco at the beginning of a word, that means muscle. That is referring to any kind of muscle. So sarcoplasm, we see sarco, that tells us it's part of a muscle. Sarcoplasm is that muscle cell cytoplasm. Remember how we looked at some cell physiology earlier in the semester? It's the same thing, but with muscle cells. As we've looked at in the tissues chapter, we have three different kinds of muscle we're going to talk about today: skeletal muscle, cardiac muscle, and smooth muscle. Our skeletal muscle. Our skeletal muscle is a voluntary muscle because we can move it consciously. But it's a bundle of elongated cells that attach to skin. They could attach to bone, or they connect to tendons that connect to bones. And these all help you move. So we looked at in the tissues chapter, skeletal muscle fibers have striations or stripes to them. These stripes, we're going to go into exactly what those are and how they work today. Skeletal muscle, of course, it can contract really fast, but just to kind of keep in mind as we're going through this PowerPoint, at least the whole first portion is all skeletal muscle. It's striated and it's voluntary. It's all stuff that we can control. It allows you to run across the street. It allows you to sit and read because your muscles still have to hold you up while you're sitting. We do have cardiac muscle. Cardiac, heart. So cardiac muscle is only found in the heart. It makes up the middle layer of the heart. When you get to the cardiac chapters and Anatomy 2, you will see exactly how that is made up. Cardiac muscle is also striated, just like skeletal muscle, but it is involuntary. You can't tell your heart to beat faster, or you cannot tell your heart to beat slower. So it cannot be controlled consciously. You can't tell it what to do. Your heart has its own conducting system, so it has its own pacemaker. This pacemaker is what helps the heart beat at a consistent rate. The last type of muscle is smooth muscle. We find smooth muscle mostly in hollow organs or hollow passageways, like your stomach. That's a hollow organ. So is your urinary bladder, because we Your airways, even your blood vessels have some smooth muscle through them. The difference with smooth muscle, it is not striated. That is where the name smooth comes from, because there's no striations like cardiac or skeletal muscle. You also cannot control your smooth muscle, so it is involuntary. You can't tell your intestines to digest food. You can't tell your stomach to finish breaking down any kinds of food. That just kind of naturally happens. So without us consciously knowing it. So that is why it is involuntary. So here's the different kinds of muscle tissue. As you can see, with the skeletal muscle, you see all these striations. This one, these lines going through. There's also several nuclei per muscle fiber. This whole portion in here, that is just part of one muscle fiber. Cardiac muscle. Cardiac muscle, they also have these intercalated discs. This is what holds cardiac muscle together. They branch, so cells can go off in different directions. And they're only found in the heart. They have the striations. You can see the striations kind of going more this way, though, in this picture. The last one is smooth muscle. Smooth muscle, as you look at the picture, there's no striations. That's what gives it the smooth look. Each cell only has one nucleus. So, and it's found in hollow organs, passageways, large arteries. It's even found in your muscles that help dilate or constrict your pupil. So all types of cardiac muscle, or all types of muscle, so skeletal, cardiac, and smooth muscle have these characteristics. Of course, they're excitable. They have that excitability. They respond to some kind of stimulus. We're going to learn how that happens. Contractility. All types of muscle can contract. I think your muscles are what lets you pick something up off of the table, pumps blood through your heart, moves food through your intestines. There you have extensibility. All smooth muscle can stretch. But if they can be stretched, they need to have elasticity to be able to go back to their resting length. So your muscles, of course, they help provide movement. They help get you from one place to the next. They help you digest food. They allow your body to pump blood around allow your heart to pump blood around your body. They maintain your posture. When you're just sitting, your their muscles are still working to keep you in whatever posture you're sitting in. They stabilize your joints. Remember when we looked at joints chapter? Some joints, specifically your knee, have a lot going on there. So there's a lot of muscles around that to help stabilize. Even your elbow, your fingers, your neck. Muscle also can generate heat. Every time they contract, they are generating heat. And that helps to keep you warm. Think when you shiver, your muscles are contracting really fast to warm you back up. They're generating that heat to warm you up. So just like anything else in the body, our skeletal muscle needs nerves, because nerves are going to tell our muscles what to do. They need blood supply. They need lots of nutrients. They need lots of oxygen to move and to produce that ATP, that energy, so they can move. They have connective tissue sheaths, and then they have to attach somewhere. Just like bone, every muscle will have a nerve, an artery, and a vein, just like bone. But in muscle, if I have, here's a muscle fiber, here's a muscle fiber, here's another muscle fiber. They're all kind of lined up nice and neat together. But every muscle fiber has its own nerve attachment. Every fiber connects to a nerve so that each fiber can work. If only one fiber had a nerve attached to it, your muscles wouldn't contract as smoothly and easily as they do when they have a nerve attached to every fiber. We need a lot of oxygen. We also need a lot of nutrients to help contract our muscles to move, to sit, to write, everything. But we also need ways to get rid of waste products. That's extremely important in our muscles. We're constantly producing waste products. We'll go over exactly what those are as we talk about how muscles work. But those nerves or those arteries and those veins, they help to pick up those waste products and get them away from muscles so we can continue doing what we want to do or need to do. How I drew those muscle fibers above, not only do each of these muscle fibers connect to a nerve, each of these nerve fibers has a connective tissue sheath around them. They all have their own connective tissue around every single one of these cells. This connective tissue is going to help support every cell. It's going to reinforce the whole muscle. Because not only the cells, but we're also going to have connective tissue going around the entire muscle. So there's different names for the different layers. Of course, just like in bone, we had different names for different layers. Muscles are the same way. We have what is called an Appy Myosum. That is our dense, irregular connective tissue, and that goes around that entire muscle. That is what I just drew in gray here. This is that epimyosum. It's going around that entire muscle. The perimyosum, that goes around each muscle fiber. Here it surrounds the fascicles. Sorry. Endomyosum is what I drew in black. Endomyosum is the black here. That goes around each fiber. Then the perimyosum is a group of muscle fibers. So here's two together. That's my perimyosum for there. There's my perimyosum for the next two. It's not necessarily just two, but that's just how I'm drawing it. So perimyosum is fibrous connective tissue that wraps around fascicles or groups of muscle fibers. That endomyosum is areolar connective tissue, and that is around each muscle fiber. Again, that's the black I drew. Here is the example from your book. You see the epimyosum, it goes around the entire muscle. We have our perimyosum. That's wrapping around this entire fascicle here. Well, endomyosum is around each individual muscle fiber. This is one muscle fiber, and that's that connective is that areolar connective tissue wrapped around each muscle fiber. So here's a chart from your textbook that shows what a muscle is, what a fascicle, what a muscle fiber is. You see what a myofibril, we'll go into detail about that. We're going to go over what a sarcomere is, that myofilament, and how these thick and thin, or myosin and actin, work together to allow your muscles to contract. We're going to go into all of that. So we kind of talked about attachments. Every muscle is going to attach to bone, skin. It may attach to some kind of ligaments that attach to bone or skin, but muscles tend to span joints. This is what allows your joints to move. If you think of that biceps brachii in your arm, it's the muscle that's in the front of that brachium, that upper arm. That biceps comes down and actually attaches below your elbow because it allows you to bend your arm. They are going to attach in two places. One place is the insertion. That is the movable part. The origin is immovable or less movable. So let's think about that biceps in your arm. I just said that it attaches, that insertion point is below the elbow. The origin is up in the shoulder. So that biceps allows your forearm to flex, to move your hand closer to your shoulder. That's what that insertion is doing. The origin, that's not going to move. That's not the muscle that lets your arm go up, say, over your head. Those are different muscles that allow us that. And again, it's all based on their origin and insertion. And as we've kind of talked, attachments can be direct or indirect. So if they are a direct attachment, that epimyosum, that outer layer of that muscle, that dense irregular connective tissue, it attaches to the periosteum of bone or the perichondrium of cartilage. And if they have that indirect attachment, that epimyosum is going to turn into a tendon or an aponeurosis. That all helps you move your joints. So let's start looking at how muscles work. And let's look at first the anatomy of each individual fiber, and then we're going to go into the sliding filament model to see how muscles fully contract. So as I've pointed out, and as that one picture above of the skeletal muscles, they're long cylindrical cells, and they have multiple nuclei per skeletal muscle fiber. They have a sarcolemma. Remember, sarco means muscle, sarcolemma. That's their plasma membrane. Just like a cell has that lipid bilayer, muscle fiber has what's called a sarcolemma as their plasma membrane. Then they also have a sarcoplasm. Again, sarco for muscle. That is their cytoplasm. Cyto is cell, plasm. That's their fluid that is found inside. Muscles contain glycosomes. Glycogen. So they have a lot of glycogen stores within each muscle cell. They also have myoglobin. Myoglobin is where we store our oxygen. Muscles use a lot of oxygen to work. So muscles tend to store some of that oxygen for when it's needed quick, because it can't always rely 100% on your bloodstream to bring them enough oxygen fast enough. Say if you are sprinting down the street at something. So our muscles will store some oxygen for use as quickly as possible to have they have that supply to start you out while it waits for more oxygen to come to the muscle cell from the blood. We have myofibrils, a sarcoplasmic reticulum, and a T-tubule. Those all are different organelles found inside a muscle fiber. So what are these myofibrils? Myofibrils, they're kind of rod shaped, and there's lots of them. So a single muscle fiber can have thousands of these myofibrils, or these rod-shaped pieces within them. Kind of like that picture above, it had all those rod-shaped things that kind of make up that one muscle fiber. This is just one muscle fiber, one muscle cell. There's thousands of these that make up one muscle cell. And that is most of that muscle cell's volume, up to 80%. That's quite a bit. So these myofibrils, they have striations, sarcomeres, myofilaments, and molecular composition of myofilaments. So let's look at the striations. We can see this is a myofibril over here. That's just one. You see all these myofibrils packed in here. This is one muscle cell with all those myofibrils. You see the mitochondria, because that makes our ATP, that gives us our energy. We have, you see the striations, you have a dark band or a dark A band. We'll go into more detail about what those are soon. And a light I band. That's what gives these muscle cells their striations, or these myofibrils. So those striations, let's look at what they are. Again, they are dark and light bands that go along the length of each myofibril. Just like that picture showed, it goes the entire length. It's dark bands, light bands, dark bands, light bands, dark bands, light bands, back and forth. Those light bands are called, or those, sorry, dark bands are called A bands. They're that dark region. Within that dark band, they have an H zone, which is the lighter region in the middle. They have an M line that goes vertically through that H zone, and it's made of a protein, myosin. And then those light bands, we said, are I bands. And within those I bands, we have Z discs. Those Z discs, they're kind of a coin-shaped sheet, and they're kind of in the middle of the light I band. So let's look. Again, we see those dark A bands. It's this dark A band in here. And right next to it, we have that light I band. And that dark A band, that light I band. Back and forth, back and forth. Those are what makes the striations. Let's look, what is a sarcomere? A sarcomere is what actually contracts in the muscle. It has A bands, and it has half of an I band at each end. We have areas between those Z discs. We'll look at pictures here in a minute. So each individual sarcomere, they align one after another. So here's the picture I was just mentioning. The I band, you can kind of see it. It goes from here to here. That dark A band. And the Z disc is the zigzag that goes through the middle of the I band. The H zone goes through the middle of that dark A band. This is what makes up a sarcomere. We have an I band with a Z disc in the middle, an A band with an H zone in the middle, and on either side of that dark A band, we have those I bands on both sides. And that makes up the entire sarcomere. And within that H zone, as you see the M line, that is found right in here, is that M line. That is all put together to make that sarcomere, because this whole H zone kind of goes from here to here, and that M line is this whole part in the middle. So let's look at what makes up this whole A band, the I bands. We have what are called actin and myosin filaments. An actin myofilament, that's a thin filament. And that is what makes up our I band, and it makes up part of the A band. They are anchored with the Z discs. So let's look at this picture. See these Z discs? these blue lines in here, those are your thin filaments or your actin filaments. They attach to this Z disc and they go into that A band. Then we have myosin myofilaments, which are your thick filaments. They go the length of the A band and they're connected at the M line. So let's look. These ones are in red. This M line in here that I colored in black that I'm not doing in red. These thick lines here are your myosin filaments. They are the thick filaments. These are part of what allows our muscles to contract. So here is a drawing of what each of these actin and myosin filaments look like. The actin is always going to be blue in the drawings, and the myosin is always going to be red. So you can see this M line. You can see how this is holding those myosin filaments all through here. This is what holds those in place. Your Z discs. This is what is holding the actin filaments in place. And these are all going to work together to help your muscle contract. You can also see these elastic filaments, these yellow parts in here. These are from titin. They are a protein that helps to kind of stabilize those myosin filaments. So let's look at how these myofilaments are made. The thick filaments or the myosin. Myosin filaments have its name because it is made of a protein called myosin. They have 2 heavy and 4 light polypeptide chains. They intertwine. As you can kind of see in this picture, you don't see it as well, but they will kind of intertwine. So there's 1, here's 2. Then you get another one, kind of going through that, and another one. So that gives you the 4 light polypeptide, 2 heavy chains. These tails, these myosin tails, kind of intertwine together. I'm going to draw this at the bottom so I have a little more room. These chains intertwining together, we are going to make, they have these club-like heads. Kind of like, think of it like a golf club. And these, then the handle of that golf club is intertwined with 2 of them wrapping around each other. If you think of it that way, it'll kind of help kind of picture how this is going to work. So when our muscle goes to contract, these myosin globular heads are going to link to our thin filament which is going to move the muscle. They're going to form what is called a cross bridge. So when these myosin heads come down and actually attach, they're going to go in a motion, the heads, these myosin heads, they'll start here and they will finish here. So they will pull this whole thin filament this way. So if you think they're starting with their head here, and their head is going to move to the position of the one right next to it, actually. And that's pulling that thin filament over. And that kind of results in a muscle contraction. So as we kind of talked about, that actin with it, the thin filaments are made of that same protein called actin. That's where they get their name. And they're technically like a kidney-shaped globular subunit. That's the best way to describe it. And they have these long chains that kind of link together and they form that actin. Kind of like I showed above. There's these chains that kind of come together. They wrap around each other and they twist together and they form that thin filament. We have tropomyosin and troponin. These are proteins that are bound to this actin filament. And they kind of help regulate some of these muscle contractions. So here's a picture that shows much better than my drawings. You see the tail, these two tails, or if you thought of it like a golf club, the two handles that come together and they wrap around each other, and then each one has its own head. These thin filaments, you see the tropomyosin as it wraps around, it is this gold strand here. This gold strand here, it's going to block. We have these myosin binding sites. That's what the kind of openings are on these actin. That is where the head, the actin binding site of this head here will attach to the actin. And it is what allows a contraction to happen. But when we're not trying to contract a muscle, this tropomyosin is what blocks those myosin binding sites. So contractions, random contractions don't just happen. It prevents just random muscle contractions that we don't want or need. Of course, we have our elastic filaments. As I showed in that picture above, those yellow at the end of the myosin, they are made of titin. That titin helps return muscles back to its normal length after being stretched. And it helps to prevent excessive stretching. We don't want to stretch our muscles too far because that could cause your muscle to actually tear. We have dystrophin. And that is what links thin filaments to the proteins of the sarcolemma. We also have nebulin, myosin, C proteins that help bind the filaments of the sarcomeres together as well. So let's look at something. Duchenne's muscular dystrophy. Duchenne's muscular dystrophy is the most common type of muscular dystrophies, and it usually starts in early childhood. Parents usually notice it because their kids get really clumsy. They start falling a lot more than they normally do or should between the ages of 2 and 7. Duchenne's muscular dystrophy, it's a sex-linked recessive disease. So it is on the X chromosome. So it comes from their mother. Males only have one X chromosome. So if they get this recessive disease, sex-linked recessive disease from their mother, they're going to get it. Females, we have 2 X chromosomes, so we're less likely to get it. This is a disease that is progressive. They start being really clumsy. Eventually they won't be able to walk. If they make it, or without any kind of supportive care, they usually do not live past 20. There is medications out there. There's physical therapy, and taking some of these medications that prevent muscle breakdown. They can live into their 30s nowadays, which is kind of cool compared to usually dying before the age of 20. So what happens in Duchenne's muscular dystrophy? They have a defective gene for dystrophin. And that's what helps stabilize that sarcolemma. So because of this dystrophin, those sarcolemmas tear very easily. We store calcium in our sarcolemma, or in our sarcoplasmic reticulum of our skeletal muscles. But if we start tearing that sarcolemma, that sarcoplasmic reticulum opens up and allows calcium to flow pretty freely throughout the muscles. And that too much calcium damages your muscles. It causes inflammation, but then your muscles aren't able to repair themselves in Duchenne's muscular dystrophy. So cells end up dying through apoptosis, and their muscle mass declines drastically. So here's a little boy with Duchenne's muscular dystrophy. He's in physical therapy, just trying to strengthen his muscles to help him be able to get through his daily life. So what is that sarcoplasmic reticulum? Again, that sarcoplasmic reticulum is that smooth endoplasmic reticulum, and it surrounds every myofibril. It runs longitudinally, so it runs along the length of that muscle cell. We have perpendicular cross channels at the AI band junction. That sarcoplasmic reticulum, as I said, it stores calcium, but it also releases calcium for muscle contraction. So it's part of the regulation of calcium inside of the muscle cells. And these T-tubules, they're formed by the protrusion of sarcolemmas. They help give these muscle fibers a greater surface area. These T-tubules have a lumen continuous with the extracellular space, so all the area around them. And when nerves communicate with muscles, it's done through electricity. So those T-tubules allow that nerve transmission to get into every muscle fiber. That's what we want when we contract a muscle. Because if our muscle fibers don't work well that way, we don't quite walk smoothly. We can't move. Our muscles are going to be disconjointed and kind of all over the place. So these T-tubules, they go into the cell at every AI band junction, and they kind of form a triad. It goes from that terminal cistern of one sarcomere, we get the T-tubule, and then the terminal cistern of the neighboring sarcomere. So here's what this kind of looks like. You see the triad, which is made up of here, here, and here. That is the white is that T-tubule I told you about. The blue on either side of that white is the terminal cisterns of those sarcoplasmic reticulum. And each part through here, those are the tubules of that sarcoplasmic reticulum that allows and helps muscles. It feeds the calcium that we need so muscles can contract. So what does that triad relationship do? They kind of act as voltage sensors. And as I mentioned before, when nerves communicate with muscles, it's due through electricity. So they're going to change their shape in response to an electrical current, which is when a nerve is communicating with a muscle. Those sarcoplasmic reticulum cistern membranes, they have different proteins. They have integral membrane proteins. So they go into that intermembrane space. If you remember from the cells chapter we looked at, they help control the opening of those calcium channels. We need calcium for our muscles to contract, so that's important. So when that nerve sends an impulse, those T-tubules change their shape. They cause that sarcoplasmic reticulum proteins to change their shape, and that's what allows calcium to flow into that cytoplasm. So I kind of talked about what happens that causes a muscle to contract. Let's look at it a little bit deeper because there's more steps to it than what I showed. I kind of showed what that cross bridge was when the myosin head's attached to the actin and the head moves from one position to another to cause that muscle to contract, but there's more to it. Of course there is. So as we're forming cross bridges and moving those actin thin filaments, we are creating tension with every cross bridge we make. As those thin filaments keep sliding over, as those thick filaments, those myosin filaments, are pushing them over. When that contraction is done, those cross bridges become inactive. They're not gonna, they won't bind anymore. So in a relaxed state, those actin and myosin filaments only overlap slightly at the end of those A bands because they need room to contract. So in that sliding filament model, so during a contraction, as I said, those thin filaments slide past the thick filaments. And that was what causes that actin and myosin to overlap. They don't change length. That actin and myosin filaments, they do not change length. They will just overlap more. So when our nerves tell our muscles to do something, that stimulation from the nervous system is what allows those myosin heads to bind to that actin, and that's what a cross bridge is when myosin heads bind to the actin. That's a cross bridge, and when those slide, that is the sliding filament theory. So let's look at all parts of it. We're going to start going through steps and what happens. So when a muscle contracts, those cross bridges, as those myosin heads attached to the actin filaments, they're going to release. The head is going to move back to its original position. So we have with my head, we have other ones kind of all along. So it's going to start here, going to attach to that actin filament. The head is going to move from here to there. The head will release from this actin filament. It'll move back to its initial position. It'll attach again and move again. That's what allows muscles to continue to shorten. Those Z discs that we looked at above, they're pulled towards the M line. That's in the middle. The Z discs are on the far outside. That's what holds those actin filaments, and it pulls the Z discs closer together. So those I bands are the white or the lighter portion. Those become shorter. The Z discs become closer. That H zone disappears. So let's look at this. We have our thick filaments in here. Our thin filaments are mostly spread throughout here. Those are the blue and the red. Then you see the titin in yellow. That is that kind of elastic type part of the muscle that helps muscles stretch, but also come back to their normal length. So these myosin heads are going to attach to the blue actin. They're going to pull these Z discs closer to this M line. These are going to go this way. So when we get a sarcomere that is fully contracted, look at how much they overlap. Those thin actin filaments are all the way to that M line. Those Z discs have moved much closer together. That H zone in the middle has completely disappeared. So there's more to it still, of course. So when our brain tells a muscle to do something, Our brain will send information down through nerves, through our spinal cord. The nerve will exit out of the spinal cord and go to whatever muscle we're trying to move. And nerve will then tell the muscles, hey, let's work. Since neurons and muscle cells are the only excitable cells in the body, they are capable of action potentials. An action potential is when a nerve communicates with something else. It is that information being sent. So, since nerves and muscles are the only excitable cells in the body, they can change. They have a resting membrane potential. They have a certain charge. And that charge can change depending on information getting sent to them or not. They have what is called a resting membrane potential. That is what their charge is when there is nothing happening in that muscle cell. It is usually between -70 millivolts to -90 millivolts. Somewhere in there. So when that nerve wants to tell the muscle to do something, nerves talk from one nerve to the next nerve, or a nerve to, could be an organ, and in this case it's a muscle. They use what are called neurotransmitters. These neurotransmitters will tell either the next neuron, the muscle, the organ, what to do. So in muscle cells specifically, there's different neurotransmitters used for different things. But in muscle specifically, they use the neurotransmitter acetylcholine. Acetylcholine, part of it is made in the cell body of the neuron. The rest of it comes from, we'll kind of learn the names, dendrites, which are the little finger-like projections that come off of the cell body. The rest of it comes kind of from that area to put together to make acetylcholine into these secretory vesicles. They use these secretory vesicles to house the acetylcholine until needs to be released out of the nerve cell and attached to the muscle cells to tell the muscle cells to do something. So we talked about ion channels when we talked about cells. Sorry. When we talked about cells, we talked about ion channels. This is where we're going to put that to use. We use ion channels to change the membrane potential. We have chemically gated ion channels. They open, especially in muscles, when a neurotransmitter connects to that ion channel. That neurotransmitter, acetylcholine, it's a ligand. Because remember, ligands, when they attach to certain ion channels, they open that ion channel. And this is what acetylcholine does in muscle cells. But we also have voltage-gated ion channels. So they're going to open or close depending on that membrane potential. Like if a cell gets too positive, because we said they're around -70 millivolts. But what happens if that cell membrane gets to, say, +30 millivolts? Well, that cell wants to be back down to its resting membrane potential, so it needs to open some of these voltage-gated ion channels. They see this 30, +30, they're going to open up to let some of this positive charges that have come into the cell out. We're going to look at how exactly this works. So these are the different kinds of ion channels. The chemically gated ion channel. In muscles, this would be acetylcholine. These green on here, those would be your acetylcholine. That is what causes this ion channel to open. Then we have our voltage-gated ion channels. These are going to open depending on the charge at the membrane of each individual cell. If you noticed this voltage-gated ion channel, it has this little ball at the end. We'll talk about that when we talk about how all of this works. Because this is what helps to participate in the cells getting back to their resting membrane potential. We've kind of been starting it, but we're going to look at the neuromuscular junction. Neuro, nervous system, muscular, muscle, junction, so between a nerve and a muscle, where they communicate. Another word that means muscles. Somatic. So, skeletal muscles, but they have somatic motor neurons that attach to them. These somatic motor neurons are what tells the muscle cells to contract. The nerves, they have an axon. That axon travels from the central nervous system to skeletal muscle. So let's see. So if I have my spinal cord here, here's my spinal cord. Nerves are going to come from the spinal cord. They're going to come out. Just a really exaggerated version of that. But this is a nerve that's coming out. Your spinal cord is considered part of your central nervous system. And when we get to the nervous system, we'll look at exactly how these neurons work to get information from the brain out. But now we're just looking at how these nerves come from the spinal cord out to a muscle. So this long section here, this is an axon. Axons can be extremely long. Think about muscle, there's muscles that move your toes. Think how long that has to be coming out of your spinal cord in your back, going all the way down to those muscles in your toes to let your toes move. These axons, redraw this, because these axons come out and they're going to branch. So here's one. They're going to come out to another one. There's another muscle cell, another muscle cell. They're going to branch into a lot of pieces to connect to the different muscle fibers. Each piece connects to its own muscle fiber here. And where this nerve, all these little projections coming off of it, each of these are going to attach to a muscle fiber. So that one may attach to that muscle fiber. This one will attach to a different muscle fiber. This one will attach to a different muscle fiber. This one will attach to a different muscle fiber. Each one of these will have its own muscle fiber that it's going to talk to. And where they connect to a muscle fiber, that is what's called that neuromuscular junction or a motor end plate. So as I said, each one has a neurotransmitter junction with one motor neuron. So each muscle fiber has its own part of that one neuron going to it to communicate. So here's another example. We show the brain, nerves come down through the spinal cord. It's going to come out and it's going to talk to this biceps brachii muscle. You can see how these nerves connect to many different fibers. There's technically one. This is going to branch out into a lot more of these actually, because we need one for every single muscle fiber. And they all need part of this nerve here to communicate with them. So where that nerve will come in, here's my nerve coming in. It's going to attach to a muscle cell. This is called the synaptic cleft. Here, this is my axon terminal. The end of the neuron is called that axon terminal. The synaptic cleft is this whole space in between. In this axon terminal, whoops, lost my entire nerve. Now my nerve is purple. There are synaptic vesicles. They are filled with our acetylcholine. I'm going to draw my muscle better here. Because in my muscle, not only do we have one fold, but we have a lot of them. Kind of go up and down like this through where the nerve comes. Those are called junctional folds. It helps to increase that surface area. And within that surface area, we have millions of these ion channels. These ion channels are these are all acetylcholine ion channels. They have little receptor sites here. Those little ligands, connection sites. Every one of these has a ligand connecting site, or also known as an acetylcholine receptor. So here's a better drawing than mine, of course. So you see all these junctional folds that come down. This is what helps to kind of increase that surface area. The purple. These are those ion channels where the green that attaches, that's the acetylcholine. That makes these ligand channels because there's those acetylcholine that attaches to this ion channel and allows it to open. The whole opening between the neuron in yellow, and the muscle fiber in red is that synaptic cleft. The end of the neuron is called the axon terminal. And in yellow, you see all those synaptic vesicles with the green acetylcholine in it. This all plays a part in muscle contraction. So when a muscle contracts, that neuron has to tell the muscle cell or send it information. So there has to be certain events that happens at that neuromuscular junction that causes that muscle fiber to get excited. And when that muscle fiber gets excited, the contraction will start through cross-bridge cycling. So we already looked at how that cross-bridging works. Now, we're going to look at how the nerve tells the muscle, hey, we need to contract. So we're going to go through these kind of step by step on how everything happens. And what I'm going to do is I'm going to draw step by step what happens here. So I have my nerve that comes out, and it's going to connect with this muscle fiber here. On this muscle fiber, I have channels. There's lots more of these. They're just kind of drawn this way. And here I also have some channels, ion channels, inside my cell. I have these synaptic vesicles. So what happens? I have a nerve impulse coming down my neuron. It is traveling from the brain down the spinal cord and into this axon terminal. Well, as this information comes down, what's going to happen is we are going to get the ion channels in the nerve activated at the axon terminal. I've got calcium out in the extracellular fluid here. As this action potential comes down the nerve, that is going to cause calcium to flow into this neuron, this axon terminal. What calcium coming into this axon terminal does is it triggers this synaptic vesicle to combine with the plasma membrane. And what happens is it's going to bind with the membrane here, and it's going to release its acetylcholine out into that synaptic cleft. The acetylcholine is going to combine. These are ligand sites They're going to combine to these ligand sites. And that's technically, because if these are technically closed until the acetylcholine binds. Oops. Bye, muscle cell. Let's redraw that quick. Sometimes too much gets erased at once. There. So once these acetylcholine binds to these channels here, they're going to open the channels. They're going to they'll open up, and what is going to flow in? We have a lot of sodium out here. This sodium is going to go in to this muscle cell. If we remember, I mentioned the charge of my cell is around negative 70 millivolts. So let me draw a chart over here to kind of show what's going on. We have our resting membrane potential. That's at that nice negative 70 millivolts. We had this action potential come. It allowed calcium to flow into that neuron. That calcium triggers the release of the synaptic vesicles to release their acetylcholine into that synaptic cleft. That acetylcholine is going to bind to these voltage or chemically gated ion channels as this, and that's going to open these chemically gated ion channels and allow sodium to come into the cell, that muscle cell. So we start at negative 70. We notice sodium is positively charged. So my muscle is resting. Sodium comes rushing in really fast. We will go from that negative 70 to roughly positive 30 millivolts because a lot of sodium comes in. I only drew a few, but sodium is constantly rushing in. It's going really fast, lots and lots of sodium. And that causes a positive, the cell membrane potential to go from negative 70 all the way up to positive 30. We're getting so many positive ions come flowing in. It's going to keep making that membrane here more and more positively charged. So what does that do? Well, what that is going to do is it is going to cause these chemically gated ion channels to close. So these are going to close off. We have an enzyme that is called acetylcholine esterase. This acetylcholine esterase is going to degrade all this acetylcholine off of these chemically gated channels. Getting rid of that acetylcholine off of these chemically gated channels is technically what closes them. When the acetylcholine esterase comes in and breaks down that acetylcholine off of that chemically gated ion channel, that's what technically causes the channel to close. I'll say it again. So we've had the acetylcholine attached to these chemically gated ion channels. That opens the channels and allows sodium to come in. When the cell starts to get too positive, acetylcholine esterase will break down the acetylcholine that is attached to these ion channels, which will cause the ion channels to close. But the inside of our cell is positive 30. We need to do something about that. So what happens is the voltage-gated ion channels that we have. These things, they're the ones that have those little, looks like a ball on a chain. Really what it looks like. These channels are going to open and potassium is going to flow out. So as potassium flows out, because potassium is also positively charged, just like sodium. When that potassium starts to leave the cell, our charge comes back down to closer to that resting membrane potential. So as potassium continues to flow out, oh, wait a second, our cell got more negative. So then what happens? Because our cell just got too negative. It's below negative 70. These little ball and chains on this voltage-gated ion channel going to come in and close off that channel. So no more potassium can go out. Our cells have leaky channels. Leaky channels allows sodium and potassium to kind of go back and forth in and out of the cell. So these leaky channels are what is going to cause that more negative to get back to its resting membrane potential. It brings the cell back to its negative 70 millivolts. But what's happening? I'm going to draw one of these channels over here, a big one, so you can kind of see. So this chain with a ball, it's going to block that channel. But the channel needs to go back to its closed position before we can receive another action potential. So while those leaky channels are still kind of working to get the membrane back to its normal resting state, these chemically gated channels are going to change position into a closed position with this little ball and chain out of the way again. Then we can receive another action potential and this whole process can start again. So let's go, let's go back here. So I have this written out for you. Action potential arrives at the axon terminal. Those voltage-gated calcium channels open. Calcium comes into that motor neuron. That calcium causes the acetylcholine to be released into that synaptic cleft, that area between the neuron and the muscle. That acetylcholine will move across that synaptic cleft and bind to those chemically gated ion channels, which opens them and allows sodium to flow into that muscle cell. That as that cell becomes more positive, the acetylcholine esterase breaks down that acetylcholine. But what happens if something goes wrong in that whole process? There's different toxins. There's different drugs or diseases that can affect that whole neuromuscular junction. If you've ever heard of myasthenia gravis, you kind of see people, they have drooping eyelids, difficulty swallowing and talking. They have a lot of just general muscle weakness. They don't have as many acetylcholine receptors on their muscle cells, because it's an autoimmune disease, and an autoimmune disease, your own body is attacking your own cells. So when that happens, those receptors for the acetylcholine are no longer there because your body has degraded them, broken them down. So your own body is attacking itself and causing all these muscle problems. So let's go back to looking at our muscle cell here. So our sarcolemma, just like our neuron, we kind of talked about it because it's polarized. It has that voltage. Where I said it was like a -70 or so, because it's more negative. The inside of the cell is more negative than the outside of the cell. And those action potentials are caused by changes in electrical charge. How I kind of showed on the last chart, on the previous drawing that I do. So we have end plate potential. We have what is called depolarization and repolarization. That was that chart that I drew out. I just didn't label it yet. So here we go. We said our cell is usually around -70 millivolts. That is its end plate potential. It has chemically gated ion channels with ligands. That is where that acetylcholine binds. That's what causes sodium to rush into the cell. As I showed those leaky channels, that's where some potassium leaks out, but not too much, because that's what causes us to go from that nice resting membrane potential. As sodium comes in, that's depolarization. Depolarization is when the cell becomes more positive. So this is where our cell is becoming depolarized. So as I drew, here's what I want. So this chart kind of explains how everything happens. This resting at membrane potential, they're showing it as -90. I says between -70, -90. So we have our resting membrane potential here. This resting membrane potential, a bunch of acetylcholine attached to those ligands on those chemically gated ion channels, and it's allowing all those sodium channels to open. Lots of sodium comes in. Our cell becomes depolarized because we have so much sodium in there. Our cell becomes so positive. Well, what's going to happen? That acetylcholine esterase comes in and breaks down the acetylcholine, which causes the sodium channels to close. But it also causes potassium channels to open. So potassium is rushing out of our cell really quick. Potassium keeps rushing out of the cell. Our cell becomes more negative, so it becomes repolarized. Then our potassium channels close. As an axon potential reaches a muscle cell, it causes a wave of depolarization. It's going to continue. I'll just draw it down here. So our muscle cell, we have all of this with where the nerve attaches, but it's going to cause, as this part of the cell here becomes depolarized, it's going to activate channels down here to open. And on this side, because it's going to travel both ways. It is going to cause these cells to spread that action potential down the entire muscle fiber. That's what causes the entire muscle fiber to contract. If we had just one small portion of it contract, we wouldn't really be able to move anything. So as everything kind of gets set out, okay, now these channels are going to open. Sodium is going to come rushing in up over here. And sodium is going to come rushing in over here. And that entire process is going to continue all the way down the muscle cell. As I drew out on the other picture, so here's, I'll do the same number they used of -7 or -90 millivolts. So we're doing fine, doing fine. Oh, sodium channels open. Sodium channels closed, potassium channels open. As I drew this part that went down below that resting membrane potential. What is happening there is called a refractory period. Remember those ion channels that I drew that had what looked like a little ball and chain on it, where that ball came over and closed that channel? What is happening during this point is this is all happening right here, because this is what is called hyperpolarization. So during this hyperpolarization point, this little ball and chain part of this chemically gated, or not voltage gated, sorry, it's a voltage gated ion channel. What is happening is that channel changes from its blocked state, so it can't allow any more potassium to flow out of the cell, but it hasn't changed its shape back to closed. So it needs to change its shape before we can receive another action potential from a nerve for that muscle to contract. So this channel has to change its shape before we can start the whole process over again. That is what's happening during hyperpolarization. And during that time, I said a muscle fiber cannot be stimulated again until that repolarization is complete. So we go from hyperpolarization back to our resting membrane potential. We need to restore that sodium potassium using those sodium potassium pumps. We need to get that sodium that came in the cell back out and the potassium that went back out back in. That's also happening during that time. That is all taking place at that exact same moment. That is during the refractory period where we cannot receive another action potential until this is done. So that sodium that came into the cell needs to go back out, and the potassium that flowed out needs to come back in. That all needs to happen during that refractory period. So I kind of look through all of this. So what is the excitation contraction coupling event? That is when that action potential got sent to that sarcolemma, part of the muscle fiber, and that triggers the sliding of the myofilaments. So that action, what that action potential is doing is it's telling those actin and myosin to work, to contract the muscle. And as I showed in that muscle, so it is propagated. So here is where that neuron attaches. But that action potential that was received right here, what propagation means is it's just traveling it all the way down along the sarcolemma and down into the T-tubules. That's what causes calcium to be released from that sarcoplasmic reticulum. And that's what allows that contraction to happen. Because we receive information, it causes all these chemicals sodium to flow in, then potassium to flow out. That all happens at this area here before contraction is even seen. All of that has ended before the muscle actually contracts, because it has to send the information down the entire muscle to cause the whole muscle to contract, not just this one portion of the muscle. So this is kind of showing we have our nerve that's attaching to this muscle cell. It's sending the acetylcholine into that synaptic cleft that is causing sodium to rush into the cell, which will cause the voltage gated channels to open and potassium to go flying out of the cell, which causes that action potential to be sent all the way down the cell. And it causes calcium to be released, which is part of what is needed, because calcium is going to bind to troponin. Troponin is this yellow piece in here. Whoops, right here. It's going to bind, calcium's going to bind to that. And that's going to cause the tropomyosin, which is that yellow strand that covers the myosin binding sites. That is what moves this whole tropomyosin that allows the myosin heads to attach. Then, as I showed, that myosin head cocks. It moves to allow that muscle to shorten. See how that myosin head here changed its shape from one position to the next position? That's what's moving our muscle along. So this is the end of part 1. I will continue recording on part 2.